You're ready to start, aren't you? Okay. How's everybody doing? How's the exam coming? So I am doing a review session. The review session is in here. I did get the room, and if somebody tries to kick us out, we know that we've got it reserved, so we can bully them around, I guess, right? Yeah. Let's see. I've got the exam written. The exam has 3 sections as before. The format's exactly the same as before. Points are slightly different. I think the first section has 16 points. The second section has 30. What is it? How's it go? 16, 45 points in the second section, and 39 points in the last section. So what happened was 3 points moved from the last section to the first section. That's the only difference. Okay. People have asked if I have am posting an old exam, and I don't do that. And the reason I don't is I really don't like to encourage you studying exams. I don't think it's a good idea. Because things change from year to year. You're much better off studying the material. So I don't provide old exams after that. The first one is just to show you the format. So you're much better off studying the material. So do that, and I think you'll be in much better shape. I think it's a fairly straightforward exam. The exam material goes from where we finished up the first exam, which was the material started with enzyme controls, and it ends with transcription. So that's the bounds of this exam. Okay? Everybody ready? Okay. So today I want to continue talking about translation, and we'll get through the process. You'll see it's a process, and yeah, there's some steps in it, and try to leave the mechanisms out as much as we can, but there's still a little bit that we go into. So I want to say a couple things. The other day when I talked about I sang you the song, one of the terms that you heard was the wobble position. And I hadn't really defined the wobble position, but I'm hoping, based on what I told you the other day, that you could figure that out. When we looked at the genetic code, and I'll remind you that the genetic code is the sequence found in the messenger RNA, and it goes from 5' to 3'. The genetic code had in that third position, excuse me, it had in that third position a lot of possible variation without any effect on the amino acid being coded for, or very little effect on the amino acid being coded for. That third position of the codon is called the wobble position. Okay? So it's able to wobble, and there's not a large effect on that. Okay? Well, the wobble position of the codon corresponds to the 5' end of the anti-codon because of the anti-parallel nature of these, right? So it's the 3' end of the codon, but it's the 5' end of the anti-codon. Okay. This shows a little trick that cells use to facilitate not having to make too many transfer RNAs. If you think about it, a codon corresponds to 3 nucleotides. And if you take 3 nucleotides and 4 possible nucleotides for each position, that means 4 to the 3rd power, and that corresponds to 64 possible codons. Well, that would mean that if cells wanted everything to be perfect, they would make 64 possible transfer RNAs. But cells go, well, why should we waste all this energy making all these different transfer RNAs? Because in many cases, in the 3rd position, it doesn't, there's a lot of variation allowed. So in many places in the transfer RNA, at the 5' end of the, 5' end of the what is this? Blasted, okay. 5' end of the anti-codon, and then this should be the base at the 3' end of the codon, all right? What we see in the anti-codon is that the transfer RNA will frequently have an I in place of another nucleotide. And the reason is that in that transfer RNA, I can pair with any of these 3 in the codon. Okay? It can pair with any of these guys in the codon. So if a cell has, if the transfer RNA has this in the proper position in the anti-codon, it only has to make 1 transfer RNA to deal with all 3 of these possible bases. So it means that cells have to make fewer transfer RNAs in order to account for that wobble position. The wobble position gives them some freedom to make fewer transfer RNAs. Okay? Okay. And by the way, I stands for inosine. It's I-N-O-S-I-N-E. And inosine we'll talk about later in the term, but I is a modified form of a purine nucleotide. Okay. Well, I talked last time about how there were really 4 steps in translation. The very first step I said was activation. So I want to start with that today and tell you a little bit about how activation occurs and what activation is. So I'll remind you that a transfer RNA has at its 3' end an amino acid covalently attached to it. And that covalent attachment is the activation process. So activation means covalently attaching the proper amino acid to the proper tRNA. All right? Well, what's the proper amino acid and the proper tRNA? Well, we know from the genetic code what the messenger RNA is specifying. So now the cell has to say, okay, well, look, here's the anti-codon that corresponds to that. I have to read the anti-codon and put on the proper amino acid that will pair with that codon. So what the cell has to do is it has to have an enzyme that actually literally reads the transfer RNA and then says, oh, this transfer RNA needs to have a tryptophan attached at its 3' end. Okay? So there's a name for the enzymes that do this. It turns out that there are 20 of them, and I'll tell you about them in a second. The general name for these enzymes is they're called amino, it's a long name, aminoacyl, A-C-Y-L, tRNA-synthetases. Amino-acyl-tRNA-synthetases. Now these enzymes are doing what I said. They read the anti-codon and decide what's the proper amino acid to put onto the 3' end of the transfer RNA. We clear on that? Now I said that there are 20 of them, and not surprisingly, there are 20 amino acids that make it into proteins. And so it turns out that there's one aminoacyl-tRNA synthetase that works for each amino acid. And yes, it's capable of reading all the different tRNAs for that amino acid. Okay? So one aminoacyl-tRNA synthetase for each amino acid, and each one of those is covalently linking an amino acid to the 3' end of the tRNA. Now I said 3' end, and you notice I hesitated because it turns out that some get put onto what's called the 2' end, and some get put onto the 3' end. We're not going to delve into that too much, but at the end of the transfer RNA, that's where that amino acid is going to be attached. Now that attachment is critical because it's the tRNA that's going to carry this amino acid to the ribosome where translation is occurring. If we put the wrong amino acid on the tRNA, then we're going to make the wrong protein. It'd be worse. It'd be just like making a mutation. Yes, ma'am.
I don't know why I got it backwards, but I think you said 5' end has the amino acid to begin with, but you're saying the 3' end?
Okay. So now let's be careful now. You're confusing the anti-codon, okay, with the place where the amino acid is being attached. Okay. So we have, if we think about the, okay, let me pull up a figure for you. So it's a good question. So let me make sure I'm clear with you here. Okay? So remember that cloverleaf structure I drew the other day? Well, this shows the thing in attempting to show it in 3D. Here's one loop of the cloverleaf. Here's the other loop. And here's the anti-codon down here. All right? There's the 3' end, all the way up at the other end of the thing. So we have a 3' end, meaning it's a physical end, versus having 5' and 3' direction down here. So the anti-codon was where I was talking about. The 5' end of the anti-codon was where the wobble was, meaning a variable base. But the end of the tRNA is here. Does that make sense? No. Okay, let me go back and get you another figure. That's fine. I want it to make sense to you. Okay, so let's go back to RNA. And oh, terrible figure. Okay, So here is what a transfer RNA looks like. All right? Here's the place where the amino acid gets attached. It's the 3' end of the tRNA. There's the 5' end. There's the 3' end. Okay? The anti-codon has a 5', we call it an end, but it's not really a physical end. It's a direction, right? 5' words versus 3' words. So on the 5' side of the anti-codon, that's where the wobble position is. But that has nothing to do with the end, that is the physical end of the molecule. Does that help? Okay. Everybody else got that? Everybody looks kind of tentative on that one. He keeps talking about ends, and there's no ends, right? There's no ends to what he has to say. I'll make sure you're clear. If you're not clear, say so, and I'll try to confuse you more. It's a joke. What's that?
The third position on the codon.
Yes. So the 3' position on the codon is the 5' position on the anti-codon, right? Yeah. I'm calling position instead of end because they're not physically at an end, right? It's the direction is what it is. Yeah?
So the wobble base is both the 3' end on the codon and 5' end on the anti-codon? Is the wobble position, or is one of them?
Okay. So look at this figure here. So this is where the anti-codon is, right? There's no end to it. We're sloppy in using the word end, but you call it side, direction, position, whatever you want. This left side will be the 5' most. This side will be the 3' most. Okay? So when I say that the anti-codon, the wobble position, is the 5' direction, the 5' most, the 5' side of that anti-codon, that's what I'm talking about. There's not physically an end.
Right, on the anti-codon.
On the anti-codon, that's right. Okay?
What function does the wobble play?
I'm sorry?
What function does the wobble play?
What function does the wobble play? When we saw the genetic code, we saw that it didn't play a very big function at all for many amino acids because that fourth position, that third position could be, in some cases, any of the 4 bases. All right? So it does perform a function because some do use that, some amino acids do use that position, but not all of them do. So we call it wobble because for most of them it doesn't matter much. All right? So a codon is 3 bases. It's just that the third base isn't as important as the other 2. Okay. All right, back to translation. Okay. All right, so now I'm going to talk about some things. I'm going to do this kind of like I did with transcription. What I've told you so far occurs in both prokaryotes and eukaryotes. Amino acids have to get activated by being covalently linked to transfer RNAs. That has to happen in both. And in fact, the overall process of translation also occurs very similarly in both, and we're going to primarily look at prokaryotic, just like we looked at before, and I'll show you a few differences of eukaryotic translation. For the most part, if you know prokaryotic translation, you're going to be in good shape with stuff, okay? All right. Well, let's start with, first of all, something that actually is unique to prokaryotes, okay? It's something that's unique to prokaryotes. Now, I told you the other day that there was a start codon. The start codon, I said, was AUG, and that start codon coded for methionine, and I told you that methionine is at the amino terminus of virtually every protein that's made. By the way, translation occurs 5' to 3', exactly like transcription occurs, exactly like translation replication occurs. Okay? So 5' to 3', everything is going 5' to 3'. Translation is going 5' to 3'. That means we start at the 5' end and move towards the 3' end. When we do that, what we have is the amino terminus, that is the amino-free amino acid, is the very first one that gets put in, and the very last one that gets put in is the carboxyl terminus of the polypeptide chain. So 5' end corresponds to amino, 3' end corresponds to carboxyl. All right. So I said that the very first one that makes it in is methionine. That's true in prokaryotes and eukaryotes, and I lied only a little bit. All right? In prokaryotes, the methionine is chemically modified. Only the very first one. Only the very first methionine that makes it into a protein is modified. It's chemically modified, and that chemical modification involves putting what's called a formyl group onto it. Okay? You can see this N-formyl group that's been put on right here. Okay? This is put on in prokaryotes, and so we call it fmet. F for formyl dash met, methionine. Now, the reason that that's put in prokaryotes is that this very first amino acid is chemically unstable. Putting the formyl group onto it protects it from reacting. And I'll explain how we get to that when we get to the eukaryotes, why the eukaryotes don't do this. But in the very first amino acid, it's chemically reactive, and it must be protected from reacting with something, and that putting the formyl group on stops it from reacting with other stuff. Okay. Well, how does the process of translation occur? Well, it's a very orchestrated process. Very orchestrated process. There are several steps to the process that we need to consider, and I'll try to spare you as much as I can in terms of detail, but there is some detail we're going to have to know in order to do this. In order for this process to occur, a complex has to be assembled. That complex includes the messenger RNA that's going to be translated and the units of the ribosome that come together. We'll see that the ribosome actually has what's called a large subunit and a small subunit. There's about 50 proteins together in the sum of the small and large subunits. A lot of proteins that are in here. This is a very complicated structure. Some of the proteins are organized into a small unit. Some are organized into a large unit. Okay. The small unit in prokaryotes is called the 30S subunit. That's what you see right there. The small ribosomal subunit in prokaryotes is called the 30S. Okay? So right now we're looking at the screen at the process of initiation of translation. This is how the process occurs, and I'm going to tell you the relevant steps in that. First of all, we need a messenger RNA to be translated. We need a small subunit. And we need some individual proteins called initiation factors. And you're going to like these. They're called IF1, IF2, and IF3, standing for initiation factor 1, initiation factor 2, and initiation factor 3. Okay? These initiation factors help to make the very first components come together. So as a result of action of the initiation factors, we see that the messenger RNA is linked to the small subunit. You can see them holding on to each other right here. You'll notice that one of the initiation factors is bringing in the very first tRNA. That's what this is over here on the side. It's bringing in the very first tRNA to start the translation process. So that happens to be IF2, by the way. IF2 brings in the very first tRNA to be translated. We can see that at this point, we've got everybody together, and we see that there's that AUG. AUG is the place that translation is going to start, and that initiation tRNA is base paired to it. There's a UAC that's paired with it. Okay? Now, there's two things about this we need to recognize. First of all, how does the ribosome know which AUG to start at? There are many AUGs in that messenger RNA. Which one does it start at? Okay? Well, it turns out that there's a sequence on here, you can see it labeled right there, called the Shine-Dalgarno sequence. This is a sequence of bases, and I'm going to give you the sequence. It's GG, A, GG. And that sequence turns out to be complementary to a sequence in the ribosomal RNA. Remember that ribosomes have both proteins and they have ribosomal RNAs. That GGAG is complementary to a sequence in the ribosomal RNA of this 30S subunit. So by pairing up the GGAG with the appropriate sequence in the messenger RNA, by pairing those together, the proper AUG is placed right here. Okay? Now, it turns out that the ribosome has 3 sites to hold transfer RNAs. They're called the A site, the P site, and the E site, ape. Okay? For the very first transfer RNA, this guy is sitting in the P site. The P site's the middle site. This is the only transfer RNA that starts in the P site. All the others will start in the A site. Okay. We're still not finished assembling everything yet. We've got to bring in the large subunit, which is over here. Okay? The large subunit comes in, and when it comes in, you notice a couple things happen. The initiation factors all get released. So that assembling of this, the initiation factors play some role in joining these guys. This actually looks like a snowman. A large subunit, a small subunit, and then running through it is the messenger RNA. It doesn't look like that here, but that's actually what happens. Large, small, and then messenger RNA running between them. Okay? Now, look at what's happened in the process here. We've done GTP coming in, and we see GDP going out, meaning that this process of assembling the entire complex required energy from GTP. GTP is the sole energy source of translation. It is the sole energy source of translation. All the energy that's needed in translation comes from GTP, not ATP. So now we have what's called the 70S initiation complex. If you do the math, you see 30S, you see 50S, and unless your math is really bad, you realize that 70S is not 50 plus 30. Okay, the S units are not additive. They're approximates. All right? So there's a 70S subunit that is the sum of the 50 plus the 30. So now we're all ready to go. We've just finished initiation. We've got the large subunit at the bottom. We've got the small subunit on top. We've got the messenger RNAs running through there. And the P site, we've got a transfer RNA with a methionine, a formyl-methionine on it. We're ready to move to the elongation phase. Okay? Well, elongation is when we're going to start adding more and more amino acids and covalently making bonds between them as they go through the process. Okay? Well, how does this happen? Here's the end of our initiation complex right here. We have the snowman. We have the initiator tRNA with the formyl-methionine sitting on there. Now we want to start making the protein. We're going to start bringing in other amino acids and linking them covalently. Well, how does the cell know which amino acid to bring in? Well, now this is where these other tRNA sites come in. All right, you see them labeled now. The P site's the one in the middle. The A site over here is going to be where the incoming tRNAs come. And the E site stands for exit. That's where they're going to exit. Okay? All right, so what happens? We see a codon sitting in the A site. That's going to determine which tRNA is going to come in. Only those that are complementary to this are going to work. All right? So let's look at this. If one comes in and it's complementary, then there's something called an elongation factor that helps the process to continue. So we had initiation factors that were IF1, IF2, IF3. In this set of the process, we've got 3 elongation factors that also help the process to move forward. They're called EF and then something, and I'll show you the somethings. Okay? The first one that's of interest to us is called EFTU, which stands for EF2, but it's not the number 2, it's the letter TU. All right? This one turns out to be really interesting, okay? What is it? You'll see that, actually it's not shown very well here, but what's happening is EFTU is bound to an amino acid linked to a tRNA. So we got the activated amino acid, and EFTU is holding on to it. Why is it holding on to it? It turns out that the bond between the amino acid and the tRNA is very unstable in water. It's very unstable in water. As a consequence, EFTU's function is, once this thing's been put onto here, is to cover it up and protect it from water. Now that turns out to be a very important function. It's so important that EFTU is the most abundant protein made in E. coli. The equivalent protein is probably the most abundant one made in your cells. If you don't cover that up and protect it from water, the water will cleave the amino acid off and translation will never occur. All right, so EFTU plays another function. Not only is it protecting, it's also checking to make sure that the base pairing is proper. Do I have the right one in here? If the right one is in here, then EFTU says, okay, I've done my job. I'm leaving. It cleaves GTP, and it goes away. Yes, this inside is protected from water also. Okay? So what we've done, we've brought in the second one. It's all ready now to join the two amino acids together. We haven't done that yet. We're waiting for that step to occur. Okay? Well, that's the next step in the process. In the next step in the process, a peptide bond joins these guys together. Look what happens. The one that was here becomes joined onto this one. This is now a peptide bond joining them. And here's something that'll surprise you. It's not catalyzed by an enzyme. Peptide bonds are not catalyzed by enzymes. They're catalyzed by what are called ribozymes. It's the first time you've heard that term. A ribozyme is a catalytic RNA. An enzyme is a catalytic protein. Now this, I'm telling you here, has only been known for about 15 years. It hasn't been known for very long. Okay? People used to think that the ribosome did all the work and all the stuff, but it turns out that the catalysis is actually done by ribosomal RNAs. The large subunit has an RNA called the 23S RNA. 23S, it's in the large subunit, and it catalyzes the formation of peptide bonds. It's actually joining these amino acids together. That's happening because, and this is called peptidyl transfer, it's happening because of the catalytic action of the 23S ribosomal RNA. Now, I'm throwing a lot of stuff at you, so I'll slow down and take questions. Yes? S stands for Svedberg units. It's actually the way people used to measure things was how fast they moved in a centrifugal field. And so it comes from that. But S, for our purposes, is just S. Yeah. Yeah?
So is that RNA being used up, or is it actually like replenished, or is it just there in the ribosome?
You're talking about the ribosomal RNA?
Yeah, the ribosomal RNA.
Okay. So his question is the ribosomal RNA having to be replenished, or is it used up or anything? It's like an enzyme. That is, it can be used over and over and over, and so it's not being used up in any way. Yeah. Yeah.
Okay, just to double check. So you have the EFTU, it protects it from water.
Yep.
And then it checks to have a proper base pair.
Yep.
And the peptide bond joins.
Okay. So it's joining the amino acids that are right there in the P site and the A site. Okay? Yes, sir.
Before it joins them, it cleaves GDP or GTP.
So So what happens is once EFTU has determined that the proper base pairing has occurred, okay, it cleaves GTP and then leaves. All right? I'll explain this business up here in a second. In fact, you don't even have to worry about this business up here. All right? Now, what if it doesn't find the right one? What if the wrong one has come in here? All right? If the wrong base pairing is in there, it means the wrong tRNA is in there, right? All right? When that happens, EFTU takes it away. So EFTU is really a quality control. It's making sure that the right transfer RNA is in the right place. Very important function. Okay? All right. No, it doesn't destroy it. It wouldn't make any sense to destroy it. It just comes back, and because it's still good, it would just be looking for another ribosome and the proper place to bring it in, right? And what's amazing, if you think about this, this whole process, folks, is occurring by diffusion. Diffusion, all right? So something has to diffuse in here. If it's the right thing, fine. If not, it leaves, okay? And we think, well, how many things diffuse in there that are the right thing versus the wrong thing? There's going to be a lot of wrong things that are going to diffuse in there, right? Yeah. But nonetheless, this process occurs at the level of about 15 to 20 peptide bonds per second. That's pretty remarkable. Think about that. How fast things are having to diffuse in and out in order for 15 correct ones to come in every second and this process to continue. Again, it kind of boggles my mind. The complexity of this system is enormous. We're talking about 50 proteins working together. We're talking about, in this case, 3 ribosomal RNAs that are there. We're talking about tRNAs. We're talking about amino acids. And this whole thing is coming together in this way. And 15 to 20 of these per second are being put together. Pretty amazing. Okay, well, we're not done. We've got to get back to where we started, right? Well, how do we do that? Well, we've got now this guy here. This one doesn't even have an amino acid on it anymore, right? So it's not of any use. We've got to squeeze it out the back door. How do we squeeze it out the back door? Well, it turns out that the ribosome shifts one codon this way. So look what's happened now. See this guy out here outside? Look where it's at now. The ribosome has shifted 3 nucleotides to the right, 1 codon. Well, movement takes energy, and the energy comes from, again, GTP. And in this case, it requires a protein called EFG, a very easy one to remember, EFG. EFG facilitates that movement and uses the energy of GTP to make it happen. Question? Okay. Pretty cool. Well, we're almost there. Now we see we've got this guy with 2 amino acids on it. We're essentially right back up here where we were with 1 amino acid on it. We start the whole process again. This time around it's going to have 2. We'll have 2 here, 1 here. Then we'll have 3, right? And just keep going around and around and around. We haven't reached termination yet. We're still doing elongation. But this process will continue over and over and over until a stop codon appears in the A site. When a stop codon appears in the A site, the ribosome knows, uh-oh, we've reached the end. It's time to stop. When that happens, we're in the final steps of translation, and they're called termination. Meanwhile, here's what's been the product of that elongation. There's that polypeptide chain. It actually comes out through a hole in the ribosome. There's a little hole that it comes out. We can see it right there. It may well have started to fold. May have started the folding process as it was being made. Okay? But now here's a stop codon that's appeared in the A site. The reason the ribosome knows it's time to stop is that there are no tRNAs that are complementary to the stop codon. No tRNAs are complementary to it. The ribosome waits. All right? It's a signal that, hey, something is happening here. There's no tRNAs that fit into here that will work. Okay? And when that happens, release factors come into play. So we had initiation factors, we had elongation factors, and in termination we have release factors. There are two main release factors in E. coli, and they do something really cool. They come in right here where that stop codon is, and they carry something very, very important to the ribosome. The important thing that they carry to the ribosome, they donate to the inside of the ribosome. Anybody know what it is? Water. Guess what water is going to do? It's going to break the bond between the amino acid chain and the tRNA. So what happens is water is donated, and look, the bond is broken and the polypeptide chain is released. We've just finished translation. We've just finished translation. Yes?
So, would chaperones be on there?
Oh, it's a very good question. Where do chaperones fit into this? Okay, so chaperones help in some cases, not in every case, but in some cases help proteins to fold properly. Okay? One of the places we find these are in the endoplasmic reticulum. Okay? So I'll actually save discussion of that for later, but I'll tell you since you've asked the question here what's happening. Certain proteins, when they're made, have a little signal on their end. At the amino end, they have a specific sequence of amino acids that tell the ribosome, I belong in the endoplasmic reticulum. It's a little signal, like a little license plate. Okay? As it is being made, this ribosome gets drug to the endoplasmic reticulum, and it starts squirting this polypeptide chain into the endoplasmic reticulum, where the chaperones will take over and help. Very cool process. Okay? Very cool process. Is that a hand over here?
Oh, yeah. You mentioned release factors. Is there names of those?
Yeah, the release factors. I just called them RF1 and RF2. I didn't give them a name, but RF1 and RF2. Okay? Well, we've reached the end. Now, basically, everybody can come apart. And there are proteins that facilitate that process. I'm not going to go into them, but they do facilitate everything coming apart. And we've completed the process. Okay.
Can I ask a quick question?
Sure. Is that the question?
Yeah, just to double check.
No, it wasn't. Go ahead.
Okay, so when the release factor comes in, it carries water to the ribosome.
Yep.
And the water breaks the bond between the amino acid chain and
The tRNA.
the tRNA.
That's right. Yep, very good. Now, the orchestration of this process is really remarkable. Okay? It's really remarkable. This process that I just described to you, it occurs in your cells, it occurs in bacterial cells, it occurs in every cell on the face of the earth. And it's virtually identical. Different names, and there's some structural things that are slightly different in eukaryotes. But this overall process of initiation, elongation, termination, and the steps that I've just described to you happen everywhere. Really, really cool. Okay, another, yeah?
How does the, is it ribosome that has the mRNA in it? How does it release it?
So, okay, so her question is, how does the messenger RNA get released by the ribosome? Is that your question? Okay, so the messenger RNA gets released by action of another release factor. I haven't given it a name. It's actually called RRF, ribosome release factor. I didn't give you that, so you don't need to know it. So I'm telling you, you don't need to know that. But there's another release factor that does that. Yeah. But it facilitates the release of the messenger RNA. That messenger RNA can go and now be translated again, right? Over and over and over.
That was my question.
What happens to it after it leaves? So if you think about this, the more this guy gets left out in the cytoplasm, the more likely it's going to be degraded. And the more it's being translated, the less likely it's going to be degraded. So the more cells have a way of keeping this thing going, being translated, the more protein they're going to make out of it. And it's a very complicated scheme that cells use that determine the breakdown rate of this. I won't go into that, but you can imagine that there's some juggling that cells have to do with that. Okay? Okay. That's cool. Well, let's see. Where are we? Many antibiotics work by inhibiting translation. If you can't make proteins, you're dead in the water. Okay? And like I said in class the other day, if we can find a difference between a process that occurs in bacteria and a process that occurs in our cells, we now have a preferential way of killing bacteria. All right? If we don't, then we can just kill any cell. We can become a bioterrorist, right? Well, one of the ways that we inhibit or we can kill cells is by using analogs of tRNAs. Things that look to a ribosome at least partly like a tRNA. Okay? One of these is puromycin. Okay? Puromycin looks very much like the 3' end of an aminoacyl-tRNA. It comes into the ribosome just fine, and when it comes into the ribosome, it gets bound to the growing polypeptide chain, but elongation can't proceed. It stops elongation right in its tracks. So puromycin works by stopping elongation. If you stop elongation, it's as good as stopping anything with respect to translation, and the cell will die. Okay? There are other antibiotics that will attack specific proteins in a ribosome and make them non-functional. And if you do that, again, you can make the ribosome itself non-functional and stop translation from occurring. Okay. You don't need to memorize that. Okay. I'll pass on that also. Okay. Let's talk about eukaryotes. Or should we sing a song first? Let's sing a song first. Okay. This is another hard one to sing, and it's to a beautiful melody, and you know I won't do it any justice, so I do appreciate your help in this one. There's a tune from West Side Story called Maria. Yeah, I'm going to butcher it, I know. All right. When I wrote this song, in fact, I used to have an undergrad who was one of my TAs, and she was wonderful. In fact, she actually wrote a couple songs. She was really good. And when I told her that I'd written this song, she says, you wouldn't dare, would you? And I said, yes, I would. She called me the bastard word, but anyway. But she liked it after I wrote. She liked singing it, but not to hear me sing it. All right. So it's called Translation. Translation. The most intricate thing I ever saw. From 5' to 3', translation, translation. The final step that we know about the central dogma. Amino, carboxyl, translation, 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 translation. I've just learned the steps of translation and all the things they say about tRNA are true. Translation to form peptide bonds in translation. The ribosomal cleft must bind to an EFTU. Translation. How high can I go? I don't know. AUG lines the fmet's cargo. 16S lines up Shine and Delgarno. Translation. I'll never stop needing translation. The most intricate thing I ever saw. Translation. Okay. All right. That was, I'm sure it was beautiful. It was not beautiful. All right. Okay. There is more junk on this floor. There's another one of these things. Okay. So, there are a few differences between prokaryotes and eukaryotes, and I've already actually told you a little bit about this already. If you think about the eukaryotic messenger RNAs, I told you that there was a difference in the structure of the eukaryotic messenger RNA compared to the prokaryotic. And the difference was two things, one at the 5' end and one at the 3' end. At the 5' end of eukaryotic messenger RNAs, there was a structure we called the cap. And I told you at the time that it played a role in translation. And the role it plays is an interesting one. At the 3' end, there's a sequence we call the poly-A, meaning that there are many A's at the 3' end, and I told you it played a role both in translation and in stability of the messenger RNA, as did the cap as well. The cap also helped protect stability, right? Okay. Well, what role do these guys play in translation. It turns out that there are proteins, one that will bind here and one that will bind over here, and the two proteins will actually interact with each other. They make a circle. They turn this guy into a circle. What does it have to do with translation? Well, without that circle, translation won't start, for one, so it's necessary to start eukaryotic translation. And second, if we think about, here is a circle now. If I start here and I move translation around over here, when I finish, I'm very close to the start again. And so by making this circle, it is thought that the translation process goes all the way around and it's able to start again very efficiently by bringing the end very close to the beginning. Because that ribosome, even though it comes apart, doesn't have to go very far to go over here and start brand new. Okay? So this circle that's made as a result of the cap and the poly A sequence, very, very important. Okay? This is what that circle looks like. No, you don't need to memorize all those proteins. All right? There's the cap. There's the poly A. And you can see all these things together are working together to get that ribosome started. Okay? So the cap and the poly A play roles in the initiation phase of translation. All right? Another important consideration is that eukaryotes don't have a Shine-Dalgarno sequence. The Shine-Dalgarno sequence told the prokaryotic ribosome, here's where to start. It said where the right AUG was, right? How does a eukaryotic ribosome know which AUG to start at? The answer is eukaryotic ribosomes start at the first AUG. They start at the first AUG. And by the way, you'll notice that the AUG is not at the end of the messenger RNA. It's internal. It's true for both prokaryotic and eukaryotic. Okay? It's internal. But in the case of a eukaryote, the very first time AUG appears, that's the one that eukaryotes use. So they don't need a Shine-Dalgarno sequence as such. Okay? The other thing that's different is that eukaryotes don't have that formyl-methionine. They don't put that formyl group on the methionine. Well, how does it keep from reacting? Okay? It turns out that the reaction happens only between the first amino acid and the second one, if it happens. Okay? Eukaryotic ribosome is structured such that they can't physically come together and have that reaction. So they don't need that chemical group protecting it. The prokaryotic ribosome will allow that reaction to happen if the formyl group is not there. But the eukaryotic ribosome is structured such that the two can't get together in the same way and have the bad reaction occur. So consequently, eukaryotes don't need the very first methionine to have a formyl group on it.
So, wait.
Yeah.
So, I'm just trying to wrap my brain around this.
Yeah. A lot of stuff today.
So, they don't have the F-methionine, and so it lets the, like, one reaction happen? Is that what you're saying?
So it allows the peptide bond to form, but the reaction that the formyl group is protecting against can't form because it's physically unable to do so. It's kept physically separated from the second amino acid's segment that would cause that reaction. Okay? So there's a physical separation in the eukaryotic ribosome that's not in the prokaryotic. So therefore, the formyl group is not needed. Yes? Okay. Never mind. Other questions? Enough mechanism for you today? Yeah. Okay, what's that? Okay, let's stop it there for today. I will futz with the camera for just a minute, and then we'll start the review session, okay?